This is Dr. Peter Larson of the Loyola Genomics Facility, and I want to ask a question. What exactly is the next in next-generation sequencing? But first, let me define DNA sequencing. DNA is a molecule made up of a long string of 4 possible molecular subunits, represented as A, G, C, or T. DNA molecules can be thousands or even millions of these letters long. DNA sequencing determines the specific order of subunits in a particular molecule of DNA. Now, one of the properties of DNA that makes life, reproduction, evolution, and incidentally DNA sequencing possible is that DNA molecules, in the presence of the right enzymes, are capable of making copies of themselves. In Sanger sequencing, the previous generation of DNA sequencing, this DNA replication reaction is mixed with special subunits that have 2 special characteristics. One, when incorporated into a DNA molecule, they cause the replication reaction to stop. And 2, they emit some form of light that can be detected. This causes the DNA replication reaction to yield a range of incomplete DNA synthesis products, each terminated with a labeled subunit. When these sequences are sorted by size, from shortest to longest, the original DNA sequence can be read by their light-emitting terminating subunits. So, what's wrong with Sanger sequencing? Absolutely nothing, really. After all, this is the technology that was used to generate the first draft of the human genome. Sanger sequencing generates about 700 letters of DNA sequence per sequencing reaction in as little as a few hours. It is limited, however, to a few dozen sequencing reactions that can be performed at once. Now, the key difference in next-generation sequencing is that some components of the sequencing reactions are fixed with substrate, like a glass slide or tiny beads. Now, when DNA replication reactions occurs, computers detect the changes to the immobilized sequencing reactants and read the sequencing reactions as they occur in real time. Next-generation sequencing produces shorter sequences than Sanger sequencing, but where next-generation sequencing obliterates the limitations of Sanger sequencing is by enabling many millions of these sequencing reactions to occur in parallel, more than making up for the shorter length of sequences per reaction. The advantages of next-generation sequencing are transformative. Sequencing the first human genome by Sanger sequencing cost about $1 billion, took 13 years to complete. Using next-generation approaches, sequencing a human genome costs as little as a few $1,000s and just takes 1 or 2 days. So, what can be done with the power of next-generation sequencing? In comparative genomics, using the complete DNA sequences from dozens, hundreds, or even thousands of individuals, researchers can link specific variations in DNA sequences with diseases, predispositions to cancers, or variations on how individuals metabolize medicines. Cancer is fundamentally a disease of the DNA, when DNA damage or mutations lead to uncontrolled growth of cells. Cancer genomics uses DNA sequencing data from cancer cells to provide insights into how specific DNA variations predict how an individual cancer might progress or how that cancer will respond to particular treatment. Transcriptomics adds an additional step to the DNA sequencing approach. RNA is first used to synthesize complementary DNA molecules, then that DNA is sequenced. The result is the complete set of RNA molecules that are being produced in a cell under specific circumstances or in response to some experimental treatment. Metagenomics sequences the DNA not of an individual, but of a community, such as the microorganisms that live on and inside absolutely everyone. This data can help identify the effect those communities have on the health and well-being of their host. And of course, this just scratches the surface of all the possible experiments that can be performed using next-generation sequencing. Talk to the LGF to see how next-generation sequencing can be leveraged to advance your biological research.